Okay, so we'll just make a start at this point. I know that mornings might be challenging for many people to be able to attend, but I want to still have this session and I want to record it, make it available to you, and we still have the evening session um, of this week. So we're on week 2 of the course, and we're looking at the chapter that deals all to do with the blood. So this is going to be the first part of the cardiovascular system that we'll have a look. And I'm just going to pull up a screen that I hope you're seeing, which just kind of gives us the similar slides that we discussed in the classroom. I'm not going to spend our time today on this session on going through each and every slide that we had a look of in the class, because you have that material available also as a full lecture recordings that I've made available on our Canvas. But what I really wanted to do was to just pick a couple of key points and spend our time and resources on going through the lab, which I assume is going to be the toughest part, especially for anyone who might have missed the in-person component of that lab. So just kind of quickly browsing through, what do you need to take away from the chapter on the blood? Like I said, this is going to be the first of our 3 chapters where we cover the cardiovascular system. After this week, we'll take a 1-week kind of break during which you will complete the exam 1 and practical 1. So that will cover the endocrine system and the chapter on blood. And then we return back to the cardiovascular system and wrap it up by having a look of the heart and also the blood vessels. So there's a lot to cover there, and all of these kind of nicely come together, I hope. So a couple of things that we discussed in the class. In terms of the amount of the blood that you have in your body, it's not really a huge amount, about 8% of your body weight, which adds up to something like 5 liters on average of an individual. So really, the blood loss would be a concern for us. What we'll notice during this chapter, or what we noticed, I think, is that blood is essential for so many reasons. And we even discussed how in clinical decision making, a huge amount of those decisions that we do initially rely on the results that we get from blood analysis. One of the pieces of literature that I was referring to quantified it as approximately 70% of the clinical decision making relying on the results from the blood work, at least partially. So clinical relevance is obvious. And then if we think about also why we rely on the blood and analysis of blood so largely, well, it comes down to the fact that accessing a sample of individual's blood is not very difficult. So drawing blood is a fairly easy procedure to do, very common, fairly inexpensive. And simply, once we've taken that blood sample and we spin it in a centrifuge, that causes the different components of the blood to separate in different parts of the test tube, and simply that already starts to give us some answers and some information about that individual's health, even before we have done any biochemical testing of the sample. So highly, highly, highly important and useful way of getting information. In terms of the functions of the blood, we often obviously end up jumping into the what's kind of what we always get taught, that it's all about the carrying oxygen to the tissues and carbon dioxide away from the tissues. Similarly, we're carrying the nutrients to the tissues and waste products away from the tissues. And then we get rid of those either through the lungs or through the kidneys and so on and so on. But there's a lot of other stuff that we might as well carry. And one of those things that I wanted to kind of highlight, tying into what we saw in last module, is going to be the role of hormones. So we never really got into the nitty-gritty of each hormone, but we got this whole big picture how hormones are the second of the two control mechanisms that we discussed that control the body systems and all other body systems. So now we're relying on the blood, and we're relying on cardiovascular system to get those hormones to all parts of the body. Again, remember that blood would reach pretty much most cells in the body. So this whole transport element is something that I think a lot of people easily can kind of get their head around. I do want to jump here and highlight the protective role of the blood as well. So we will actually spend 1 week talking about the immune system in a greater detail, but it's the immune system that works through the white blood cells. So you'll be introduced to the white blood cells in this week's material, but then we'll dig in a greater detail to that later on. In terms of if we think of the protective functions of the blood, I think blood clotting is a good one as an example as well. So we get all sorts of cuts, might bleed, whether it's internal or external, but your body really has a special component of the blood that plays role in making sure that those bleedings, those cuts, do not get to carry on. So we make sure that blood starts to if it's an external cut, we form a scab and stop that loss of blood from the system. The final one that I've kind of grouped here in terms of the functions, the regulatory function of the blood. Well, I think you could throw hormones here as well to the regulatory function. So it's a kind of a wherever you wish to place it. But some other examples that I thought of, and there's a lot more that we could add here, is that you remember when we talked about the basics, fundamentals of life, the importance of water as the foundation of all living things. And we saw that there might be times, there might be needs when we need to adjust the amounts of the water or amounts of the fluids at different parts of the body. So that's where the blood becomes especially useful and especially important. So we're able to move fluids from one body compartment or one body part to another really easily. Majority of the blood is, after all, water. The other thing that we notice that is that blood itself, just since it is largely made of water, and we discussed the whole concept of living by the beach, how big bodies of water have this amazing ability to absorb a significant amount of heat during the day and then release it over the day, which kind of evens out the fluctuation of the heat at the coastal area. That's why coastal areas are nice to live on. Similarly, blood, being largely of water, is able to transport heat from one body part to another. If you think about it, for example, going to freezing temperatures where we would expect that you would get a frostbite to parts like tip of your nose or your ears, that doesn't happen as quickly as we would assume, simply because of the fact that we're able to transport heat to these body parts with the blood. Also, regulation of body pH. Blood pH is maintained somewhere around 7.35 to 7.45 typically, which is a really narrow margin. So again, it's a great tool for us to make sure that the body pH is maintained at the optimal conditions, just slightly alkaline. But I guess that having seen this, the big message that I wanted to put across is that blood is important. We see that, or if we remind ourselves that blood is a special type of connective tissue, we revised different tissue types already earlier on your studies, and blood was the kind of odd man out from the connective tissues. Here we are seeing kind of a nice listing of everything that blood is made of. So, like I said, we can draw the blood sample. Here we have a test tube where we've drawn the blood sample, and then we put it to the centrifuge where it has spinned real fast, and the spinning motion has separated different elements to different parts of the tube based on their weight. And this allows us to have like a very basic understanding of what blood is made of. So majority is going to be plasma, so just slightly over half of the total volume of the blood is going to be plasma, and we'll talk about that a little bit more. And then everything else, you can think of plasma as kind of the liquid matrix where everything else is in. So everything else is either cells or components of the cells or fragments of the cells, and they are called formed elements. So formed elements are this kind of floating about in the plasma before we separate these. And majority of the formed elements, so about 45% of the total volume of the blood is going to be our red blood cells, our erythrocytes, and then small amounts of white blood cells, also known as our leukocytes, and platelets. Each one of those two represent about less than 1% of the total volume of the blood. And what we'll see that they form the white blood cells and platelets, this kind of a buffy coat layer between the plasma and the red blood cells. In general, if we think of the blood, there is a limit to the life cycle of the cells within there. So there's a constant process of removing some, adding some. So that's something that we'll keep seeing. And that actually brings us to talk about some of the terminology that I highlighted in a class. And if you've taken medical terminology, this will be very familiar for you. So when we talk about poesis, that refers to formation of something or making of something. And when we talk about different components of the blood, we talk about different types of poesis. And this is where learning the terms once becomes so important. So we'll see that the general term when we talk about blood as a whole is gonna be hematho. So hematopoiesis would refer just to production of the blood in general. So when we're talking about that, you're already able to make that connection. Erythropoiesis then refers to the making of the red blood cells. Remember, these were our erythrocytes. And finally, our leukopoiesis refers to the making of the white blood cells. So we'll talk about each of these real fast a little bit, but at this point, I guess that the important message to take away from that is that learn the terminology once, it will make your life easier. Here we have the same components that we saw earlier on this test tube that I have still left on the right-hand side of the screen, but they're spread apart on a microscopic glass slide, so you can see all of them on a microscopic view. And what we'll see is that the red blood cells, these nice rounded circles that seem to be a little thinner from the middle, these discs, they form about half of what we can see in this field of view. The space between all the cells that kind of looks pale on this, that would be the plasma. So we just can't see it super well, but that's where everything else is suspended in. We can also see white blood cells that were located on this buffy coat as these larger than red blood cells, larger cells that have a darkly staining nucleus on H&E stain. And then if we look very, very closely, we can see odd little tiny drops there. They are platelets, so fragments of the cell. What's important to take away from this is really to just focus on the proportions of how much each one is present. And that aligns with what we saw here on this larger vial of blood sample that had been separated with the centrifuging it. We're seeing the same thing on a microscopic level. So I guess that's the kind of the first big thing to notice. A quick run through different cell components of the blood, since we have a limited time today. Let's start with the plasma. So plasma is this kind of, if I were to describe it, little bit of yellowish or straw colored liquid, about 90% water. And then we have a bunch of dissolved materials. These are largely proteins. So some of the proteins that are going to be important are going to be albumin, so important carrier, buffer, regulator of the osmotic pressure. About 60% of all the proteins in plasma are going to be albumin. Then we have globulin that was important in transportation of materials, immunity, and so on. And then fibrinogen that plays a role in clotting as well. We do have other materials that we just discussed earlier on: nutrients, gases, hormones, wastes, and then a kind of a group where I put the rest inorganic ions as well that we're carrying with the plasma. So a lot going on there, but really, if you want to simplify, maybe a little oversimplify, it is this kind of a sticky-ish, thicker than water in its appearance, in its viscosity, fluid where everything else, all those formed elements, are then located. And what I wanted to do next was just to go through the remaining parts of the blood. So like I said, they are either cells or parts of the cells. So they're going to be our red blood cells, white blood cells, and platelets. And we'll just quickly run through those. We already discussed about the term poiesis. So making of the blood is largely thanks to the process assigned to the bone marrow. So that's why bone marrow is super important. And when we talked about the skeletal system on your BIO201, you remember skeleton does so much more than just give a structural support and attachment points for muscles and so on. So our red blood cells, the term that I'm expecting us to use is erythrocyte. That's the proper scientific and medical term. So again, going to the making erythrocytes, that's gonna be erythropoiesis. If we look at the shape of the red blood cell, it's kind of this flattened disc, slightly embedded from the middle. So the term biconcave disc refers to this, that it's a disc shape and it's concaving in on two sides. So it's biconcave. Red blood cells are anucleate, and they have no cell organelles. So no nucleus, no cell organelles. And that brings us to wonder, how is this possible, since you've learned that cells do typically have a nucleus and they have cell organelles? Well, this was the case at early parts of the development of the red blood cell. But as time goes on, the nucleus and cell organelles get removed. And this actually can be explained, but when we think of why would this happen, can be explained by the function of the red blood cells. The main function that we associate with the red blood cells is the transportation of oxygen bound to the heme groups. So we're eliminating the chance of using extra resources by eliminating these cell organelles. So we really are doing a good job on transportation, not sacrificing some of the material that we're carrying simply because of the cell actions that take place. Actually, the shape of the cell that I mentioned earlier on, this biconcave disc shape, is ideal for oxygen transport. And this is where hemoglobin, which is often abbreviated in medical records as HB, becomes very important. About 97% of the cell volume without water of the red blood cell is hemoglobin. And hemoglobin is made of this red pigment, iron pigment, and then the globin, which is the protein that these are bound to. We end up noticing that males have a little bit more hemoglobin on average on their blood, between 13 to 18 grams per 100 mils, whereas females are somewhere between 12 to 16 grams per 100 mils of blood. This is where also the hemoglobin and thinking about the hemoglobin becomes important when we notice that it is responsible for carrying the oxygen molecules, but carbon monoxide can bind to it, and it binds to it with much greater affinity, which results in the oxygen atoms being blocked from binding. So that's why carbon monoxide poisonings are such a big issue for us. Each of the hemoglobins carries 4 oxygen atoms, and if we look at the color that hemoglobin then gives to the red blood cell, this color is going to be really useful for us, especially when we get to talk about the blood vessels. So oxygenated red blood cells, so red blood cells that carry oxygen from the lungs to the tissues, they're going to have a kind of a more of a brightish, reddish color, whereas the deoxygenated red blood cells that have given off the oxygen, for example, to the tissues in the body and are now returning back from the systemic circulation towards the heart, they have a darker color. It's not quite blue like you often see us using in textbooks, bright red and blue. It's more of a purplish color, but there is definitely a color difference. In terms of the size of the red blood cells, they might be just slightly larger than the smallest blood vessels, smallest capillaries in our body, because they can slightly squeeze through the narrowest vessels. Red blood cells are produced at the bone marrow, and we can have issues if we have too much red blood cells, so hypoxia, or too little. Let me correct, if we have too little red blood cells, which would be an example of hypoxia, or if we have too much, that will have an effect on the viscosity of the blood. But really, the making and removing of the red blood cells should be in balance. So we should be putting out same amount as we're removing from the blood to maintain the homeostasis that we discussed about in last session. A couple of hormones, or one hormone that I especially want to mention relating to our endocrine system is going to be our EPO, so erythropoietin, important hormone regulating the production of the red blood cells. We have some examples of what could cause EPO to be produced in higher amounts, issues either with condition of you or at certain illnesses or living in higher altitude. And this is sometimes misused by athletes because the logic is that if you can get EPO released to your body, that triggers more production of the red blood cells. And if you're in the very high level athlete world, the smallest of advantage can be the one that defines whether you win or not in a competition. So if we have two individuals that are perfectly equal both in all other aspects, but one of them has more red blood cells, theoretically, this individual is now able to carry more oxygen to their tissues, so they have a greater chance of succeeding on that that particular competition. So it would be ideal in that sense for that individual to have more red blood cells. So some athletes whereby trying to bring up their red blood cell count by getting the EPO hormone up. And one way that we see a lot of athletes, and that's a completely acceptable way, is to go before important competitions to high altitude training, spend time there, because as a response to lower oxygen concentration in the atmosphere of the high altitude, your body will just naturally start to produce more EPO. Well, that would be acceptable, but you would not be allowed to, for example, inject more EPO to your a body. That would be considered as doping. So there's a little bit of balancing there. What I wanted to talk just a little bit about was, before we jump to talk about that, just remind ourselves that the lifespan of a red blood cell is somewhere around between 100 and 120 days. They eventually get broken down into various different components that we can then reuse. But I wanted to talk about this condition, and this is a blanket term, disorder called anemia, which really refers when we have too little oxygen available. And you get all these signs and symptoms that align what would happen if you're not getting enough oxygen to the tissues. So tiredness, pale, cold, things like that. And again, there might be many different reasons for that. We might be losing blood. Losing of the blood, bleeding is easy to notice if it's external. If it's internal, that becomes a little harder, and that can go on without us noticing it immediately. So that's a big concern for us. One big problem would be if not enough red blood cells are being produced, and we just talked about the role of the EPO on that, or if too many red blood cells are being destroyed. What happens when we destroy red blood cells is actually that after about 100 to 120 days, we break the red blood cells into their components, and these components can be then reused to build future red blood cells. So we get our heme, iron, and globin elements that can be reused. But I wanted to go to a special kind of example when we're talking about anemia. And I want us to look at the sickle cell anemia. So here I have an example of a typical red blood cell, what it should look like, and a sickled red blood cell. So this kind of a crescent moon-shaped red blood cell that results from just one base difference in the genetic code that codes for the red blood cell, which causes one amino acid difference. And you'll see how smallest of difference in the chemistry, chemical structure, can have a huge effect on the physical appearance. So the condition that I wanted us to especially draw our focus on is this sickle cell anemia. And what happens because of this mutation, the red blood cell is wrong shape, and now this can cause issues, blockages in the vessel when this nice rounded shape doesn't flow past the smallest of vessels as easily. It also has an impact that there is a weaker oxygen delivery in these sickled red blood cells. What we notice, what I think is rather interesting, is that this is mostly affecting individuals that are from or whose ancestors are from sub-Saharan Africa. And you might initially think that that's really unfortunate for these individuals. How did this happen? But we have to always take a little bit of a step back and remember that evolution maintains certain characteristics for a reason. There is another condition that we see on this same area mapping nicely, which is going to be the prevalence of malaria being especially common in the areas where sickle cell anemia is common. And a malaria parasite requires as one part of its life cycle to enter to the red blood cell where it multiplies and then breaks out and spreads everywhere. It needs this access to the red blood cell. And if the red blood cell now is sickle shaped, it's not able to do that. So actually, you could basically, if you stretch the lines, so stretch the story a little bit, almost argue that sickle cell disease gives you protection, or has a protective function against malaria. So that might be a good example of showing how evolution is not foolish. We always have a reason. We might not, we humans, immediately pick it up, but there's some sort of a bigger plan why certain traits are preserved. If individual has sickle cell anemia, we can do transfusions. We can also use various different kind of chemicals that prevent the sickling of the cells, red blood cells, and so on. But this question talking about the anemia in general, and then sickle cell anemia. What is sickle cell anemia? What causes it? And what do we see there? And what kind of consequences this odd shape has? And then relating it to another condition where it's common and another condition where it's common. This would be a very good extra credit question in your exam. So a little mini essay about that. And if you feel confused, we can come back to that when we revise for your exam next week. But I want to put it out there that that is probably what I'm going to be asking as your extra credit question in the exam. Let's have a look at the next group, which is going to be our white blood cells. So the proper term to use is leukocytes. You remember they are not as numerous as our red blood cells, about less than 1% of the blood volume. So in this field of view of the blood smear that we see, we only see about 2 white blood cells. White blood cells are characterized by a little larger size if we compare them to the red blood cells on a microscopic scale. They're kind of rounded, big nucleus that's very prominent with the staining of with the H&E stain, like I said. Actually, white blood cells do have nucleus, which, like I said, is prominent, and also organelles. Their main role, if we kind of simplify, is to do with the disease defense, defending us from diseases. And there's going to be 5 different kinds of white blood cells that I want us to get familiar with. And once we go through those, the easiest way to go through them is to separate them into 2 groups. Granulocytes, which means that we see clear, obvious granules in the cytoplasm of this type of white blood cell, and agranulocytes, which have a cytoplasm that's less distinctively granulated. So it's kind of a smoother in appearance. So let's have a look of these white blood cell types. So we're going to go through all of them in the practical for this week. I would expect you to be able to identify them, maybe remember some of the functions as well. In the exam, we won't go into that much of it. Let's start with neutrophils, the most common of these white blood cell types that we're looking at. We see those granules in the cytoplasm, and we see that it has multiple lobes for the nucleus as well. Neutrophils play a role in the immune defense reactions. And the next one that I wanted to talk about is going to be our eosinophils. Eosinophils are second least common, so not very, very common at all in the white, when we count the white blood cells of the blood in a normal stage. What we see, again, that granulated cytoplasm, and we can see that the nucleus has 2 lobes, so it's called binucleate. Eosinophils play a role against parasites and allergies, allergic reactions. And then the last one from this granulocyte group is going to be our basophils, the least common type of the white blood cells typically. Basophils, again, we see those granules in the cytoplasm, and now the nucleus is really taking all over the cell. It is kind of dotted in appearance, so helps us to identify playing role in inflammations and allergic reactions and so on. The two remaining ones, if we look at their cytoplasm, the cytoplasm is much smoother than what we saw earlier on. First one, lymphocytes, they are actually our second most common white blood cell type of these that we see. No granules in the cytoplasm. It kind of stains blue, the cytoplasm as well, if you look at that. One large nucleus that's present there, but doesn't take quite the same all over the cell as the basophils appearance. Lymphocytes produce antibodies, so you remember our T cells and B cells, T cells maturing in thymus and B cells maturing in bone marrow, being important for immune responses. The final one, another agranulocyte, is going to be our monocytes. Our monocytes are, again, no granules in the cytoplasm, and the nucleus, if you look at it, it's kind of a horseshoe shaped. These are important cell eaters, so phagocytosis process engulfing an invading cell, for example, if we need to get rid of that. So I guess that a good question that I could ask from you in the exam would be, for example, can you list the different white blood cells in the order of from most numerous to least numerous? And I would give you the names of the white blood cells. I would just simply ask you to be able to put them in a list in a logical order from the most common one to least common one. And as always in anatomy and physiology, try to use mnemonics to help you. So the one that I have thought about for here is neutrophils, lymphocytes, monocytes, eosinophils. Basophils is the order. So how about never let monkeys eat bananas as a reminder of that order? Of course, we have to remember that although white blood cells represent a small amount of the blood volume, less than 1%, and different types of white blood cells had differences of how common they are. If we have an infection, we end up then producing, as a response to that, more white blood cells. So the number of white blood cells grows. So the white blood cell count increases in response to infection. Even though we don't know where in that patient the infection is, it gives us a clue that there is an infection. So it's kind of a handy first tool for us. And we'll get to that in greater detail when we talk about the immune system and immune responses later on in a few chapters' time. I'm thinking if I should say something about leukemia. I'm going to skip it and you can watch the full lecture video for having a little bit more of a detailed story about that. But what I do want to mention is the platelets. That's the final cell component, final type of formed element that we haven't looked at yet. And it might be hard to see on this picture, but they actually do still show up on microscopic level when you observe the blood smear. They are tiny, tiny, tiny dots. These are not full cells. They're actually fragments of cells, and they play an important role in clotting reactions, so forming those clots. The life cycle of a typical platelets is fairly short, about 10 days or so. And I think that that's a good review of everything that I really wanted to say about the overall quick review of the blood. What I'm gonna do, I'm gonna next share with you the screen of my, let me just change that, of my Canvas course. And remind you of what I'm asking you to do this week. So for this week's assignments, you have a quiz and you have the practice anatomy activity to do. So the quiz, again, remember that I'm not asking you to be experts of everything that's in the quiz by the time you take it. Quiz is a learning chance. So while you're working through the quiz, refer to the textbook. There's on the left-hand side of the screen on the quiz page a link that takes you to the textbook, and you can check concepts that you're learning about. So that really is a part of the learning process when you work through the quiz, to do the reading of the textbook, pull the answers from there. And as you'll notice, blood quiz carries quite a bit of points towards your final grade. So it's worth of doing. The second element that I'm asking you to do for this week's labs or this week's homework is going to be the blood anatomy practice. So again, it's going to be pictures. It's going to be structures that I'm asking you to identify, tell things about, and so on. And both of these are completed on Canvas. The anatomy practice is a very worthwhile activity to do because that prepares you for that practical, that first practical that covers the endocrine system and the blood that you'll be taking as part of your exam week next week. I will make sure that you're all set for success for that. You'll get a chance to practice practicals and so on before you take that. But I just want you to know that that's you doing that work ahead of the time, preparing for the exam or the practical test. The final thing that I wanted to discuss about today refers to our lab, which was completed in a class. So we did some blood typing and considered some transfusions. What kind of blood can you give and to who you can give it to? This was an activity like all of our labs that I'm asking you to be in person to complete it. So there is an element that you needed to be present. If you were not present in the class, you really should be reaching out to me. Let me just stop sharing. I'll update that link. Just a second. So what we see is that when you click onto that activity, I'm trying to pull up a right window. Bear with me. When you go to that activity, you will find a worksheet that I'm asking you to complete, and this counts as your lab report for this particular lab. And the first 4 questions are a little bit more of a dry lab type of thing, so you work through them based on everything you learned about blood types and matching blood types and all that. We'll go through that together also we went through that in the class while we were kind of practicing that. But the last 5th question is you actually working out your own blood type. And for that, we use these little blood test cards that you completed. You drew blood from yourself a little, and you use the antiserums to notice that what kind of a blood type you might be, and defined your ABO and Rh positive or Rh negative group. So the big thing to remember from this was that if we had the agglutination reaction taking place for anti-A serum, you were then the blood type A. If you had agglutination for anti-serum B, you were blood type B. If you had agglutination for both, you were then AB. If you had no agglutination reaction for anti-A or anti-B, you were blood type O. And finally, if you had the agglutination for anti-D, that means that you had D antigens on the surface of your red blood cells, which means that you are Rh positive. Alternatively, Rh negative if there was no reaction for that anti-D. I don't expect that you have to draw the card. You can take a picture of it and upload it with your lab report. That would work just fine. But I want to see that you're able to work that out yourself. If you were not able to make it to the class, contact me and I'll give you advice what to do about this question. But I'm expecting that if you were in a class, you include that. This table and the various questions that I provided about the blood typing, I think we went through them pretty well in the class. And just for those, if there's anyone who missed that session, I'm going to give a really quick run through of what sort of things to think about to succeed on that. And again, I just have to remind that we're focusing on ABO blood grouping and Rh positive, Rh negative. We're not looking at the many other blood grouping possibilities that we have. Even though you can work as a group on your lab benches, I am expecting an individual submission from each member. So you should be uploading your answers to the Canvas. So we saw that when we talked about the history of a blood typing and all that, that sometimes blood transfusions, when we first started looking at those, when they started to be done in 1900s by Karl Landsteiner, that they could save someone's lives, but sometimes they also were the death of someone. So clearly there was something that was important, that you could give blood from one individual to another individual, but it did not always work. And that work actually got Karl his Nobel Prize in 1930s. There are about 30 other blood types. They don't cause that much transfusion reactions, so we're not going to be focusing on those. But what we covered in the class while we were kind of preparing for this lab was that, first of all, we remember that you could have 4 different types of blood types when we talk about ABO system. You could be blood type A, in which case you inherited from at least one of your parents the allele for A antigens on the surface of your red blood cells, or you inherited it from both parents. So here we have example of those A antigens on the surface of the red blood cell. I think of them with the analog that I gave you, to think of donut as a red blood cell and then the A sprinkles on the surface of that. So those A sprinkles would be the antigens for A. Blood type B similarly has to inherit the allele for blood type B antigens, the antigens on the surface of the red blood cell from at least one of your parents or from both parents. Only way you can be blood type AB is if you inherit it from one parent an antigen, or the allele for antigens A, and from one parent an allele for antigens B. So you would have both of these antigens on the surface of your red blood cells. And the last one was blood type O, where there's actually no O antigens. It just means that there's nothing. So we call it blood type 0 in many other countries, but we don't do it here. But we call it blood type 0. So the only way you inherit that is if you receive a recessive allele from both of your parents. So here we have actually a better look of what the actual antigens look like in diagrammatical sense on the surface of the red blood cells. Of course, the scale is a little off. And what we saw is that if you had antigens on the surface of your red blood cells and you received blood that had antibodies against those antigens, then there would be a agglutination reaction. So whereas normal blood smear looks like this, you have the red blood cells nicely spaced apart. You can see some white blood cells and platelets there too if you're very observant. This is what we're expecting to see. If there is a reaction because your antigens and then the antibodies don't match. So you've now been given blood where there's antigens on the surface of those red blood cells that react with the natural antibodies that you have in your body, in your blood. There will be a agglutination reaction. So you see how the red blood cells kind of tend to clump together, and this would be an issue in many ways. So we don't want to see this. And actually, when you completed your blood test card and you use those anti-serums, if you had the antigens and antibodies causing agglutination, you could see it with a naked eye. And I'll add a link to a video where you can see me actually doing this in a office. So to the end of this video as a link, if you didn't have a chance to attend to the lab, you can see what's going on there. So there I simply have the antigens and antibodies reaction being shown. We completed together this chart where we saw that if your blood type was A, you had those A sprinkles, so antigens for A on the surface of your red blood cell, but you had also anti-B antibodies in your blood. So if you have antibodies for B, you will cause a reaction if there's any B around. So you should not normally have B antigens in your blood. And if we find blood transfusion where we all of a sudden get B antigens, these antibodies will attack them. So you can give blood from donor whose blood type is A to an individual who is blood type A. If the individual, the recipient, is blood type A, and they receive blood from a blood type B, there would be a reaction, agglutination, because now the anti-B antibodies on this recipient's blood will attack with the B sprinkles, B antigens on the surface of the red blood cells of the donor. Same happens with if the donor is AB, because the A part would be okay, but now the B's on the surface of the red blood cells from the donor, the B antigens, cause a reaction with the natural anti-B antibodies on the recipient's blood, and there is agglutination. If you receive blood from an individual who's blood type O, well, that means that that individual who donates you this blood is not going to have any antigens on the surface of their red blood cells. Remember, it was blood type 0, 0 antigens. There won't be anything for your antibodies to attack, so that reaction would be also fine. And we can work in the same way through the entire table. And what we'll end up noticing is that individuals who are blood type O can donate their blood to anyone. So they are called universal donors. And individuals who are blood type AB, they can receive blood from anyone. So they're called universal recipients. And having said all that, I think that the most important, we've covered the most important things about the ABO system, and we just need to add one level of complexity to this discussion, whether you're Rh positive or Rh negative. And there are many Rh factors. The most common ones are C, D, and E. And it actually turns out that about 85% of Americans carry D antigen on their red blood cell surface. And that means that they are Rh positive. This would not be an issue if you have a negative match, or you have a match where these don't, or you have a transfusion where these don't match, unless there's been a previous non-match and we have now had a chance to form antibodies in response to that previous exposure. So first, blood transfusion might not be a problem, but the second one would be, because now we have these antibodies to attack something that's not a match to us. Same goes on with pregnancies, giving birth. I've never seen a birth where some of the blood of the mother and child wouldn't mix. So what ends up happening is that first pregnancy, if the blood types for Rh are not a match and there's mixing of the blood, that's fine. But now the mother has developed antibodies against of what they were exposed. So second pregnancy would be a problem if the mother and the child do not match with their RH. This is what you did in the class. So you saw if you had a reaction for anti-A serum, but you didn't have a reaction for anti-B serum. That meant that you were blood type A. If you had a reaction for anti-B serum, but not for anti-A, that meant that you were blood type B. If you had a reaction for both A and B anti-serums, that meant that you were AB. And if you had no reaction for A or B anti-serums, that meant that you were blood type O. And by reaction, I mean this agglutination where we can see this clumping of these red blood cells that we had a look of just a while ago on this diagram, this agglutination reaction. So that allowed you to work through your ABO blood type. And then the last thing you needed to add to that consideration was whether you were Rh positive or Rh negative. If you were Rh positive, you had D antigens on the surface of your red blood cells. So positive will have agglutination reaction in response to this anti-D serum. So I'll just leave that and have a look of some of the questions that we had for the lab. We remember if you're blood type A, so you will now have A antigens on the surface of the red blood cells. And similarly, you will have these D antigens on the surface because you remember Rh positive always had Ds on the surface of the blood cell. In terms of the antibodies, remember these are what dealt with if there's a pathogen in your body invader. You're constantly looking, since you naturally don't have any B antigens, you're looking for B antigens through the antibodies. So your antibodies are anti-B in the plasma. For A negative blood type, you will have those A sprinkles, so the A antigens, but you will not have D sprinkles because you're negative, not positive. So the antibodies that your plasma will carry are anti-B and anti-D. For blood type B positive, you will have antigens B and antigens D. So B for those antigens that make you blood type B, and D meant that made you Rh positive. So that what you're screening against naturally is anti-A, and that's why you have the anti-A antibodies. Same goes for B negative. So you will have naturally those B antigens, but you will not have D or A antigens. So your antibodies in plasma are going to be anti-A and anti-D. AB positive will have all of these antigens on their surface of the red blood cells. So you have the A and B antigens that makes you AB blood type. And then you had the D antigens, what made you a positive for Rh. So you will not have any antibodies for these on your plasma. AB negative, you have the A and B antigens, but you don't have the D antigens because it's negative now. So the antibodies that you will carry are anti-D antibodies. Blood type O means that you don't have A or B antigens. It's the plain donuts, plain red blood cells. You will have the D antigens because you're now Rh positive. So what the antibodies that you carry are going to be anti-A and anti-B. Blood type O negative, well, you don't have then any antigens of these ones that we've been looking on the surface of the red blood cells, but you will carry all of the ones that we've been looking of the anti-antibodies. And similarly, we had a look of agglutination reactions in many different scenarios. and I'll leave that for you to have a look. We had a look of recipient donor matches, and again, you can have a look of that. And we considered that also when it comes to the plasma transfusions. But what was important for the question 5 was to have that your own blood typing card. And remember that I was very happy to receive, alternatively, if you didn't want to draw or you could take a photograph of your blood typing card and attach that with your lab submission. Of course, I'm also expecting that you have identified your own blood type from that. Make sure you submit this by the end of the week, like all other assignments as well. And that should get you completed with the lab. So at this point, I think that we've covered quite a bit of material. We've covered everything that I can think of that you need to know to succeed in this week's assignments. If you have any questions, if you can think of anything that I didn't get a chance to touch upon and we haven't talked about, do not hesitate to reach out to me. Remember that I am here to help every step of the way. And we will meet again on a virtual format tonight at 8 o'clock if you want to join me. And until then, I thank you for your attention and I hope you have a very good day. Thanks. Bye.